Welcome to the United Leukodystrophy Foundation presents LBSL by Dr. Amena Smith, Dr. Christina Mertz, and Dr. Marc Engelen.
Good morning. This is Ali Fatemi from the Kennedy Krieger Institute. I'm a pediatric neurologist here who has been working on leukoencephalopathy, brain strain, and spinal cord involvement and lactate innovation now for a few years. I was going just to do a brief introduction of the speakers, and then I'll go into the background. Before we go there, I just wanted to briefly mention that the disease is a new disease, relatively new, discovered about 15 years ago by Professor Van de Knaap from the Netherlands. I vividly remember the first time when she presented this disease, when she had the first set of cases. I believe there were 7 cases, where we still didn't know the gene. It was then eventually published in 2005. When the first paper came out, and shortly thereafter, it was found out what was the responsible gene for this disorder. And I think we really thank Dr. Van de Knaap for all the work that she has done in that realm. And we have tried to sort of continue that pattern and have now a collaboration that's cross-Atlantic between the universities in Amsterdam and us at the Kennedy Krieger Institute. So therefore, the speakers today is a mix of Dutch and Americans. The first speaker is Dr. Marc Engelen, who's a pediatric neurologist at Amsterdam University, part of Professor Van de Knaap's group. Dr. Engelen is very well known, obviously, in the adrenoleukodystrophy field, where he's a very established expert, and has also become interested now recently in LBSL and is working with us on clinical studies. Then we will have Dr. Christina Mertz from my lab at the Kennedy Krieger Institute. She's a scientist in our lab, a neuroscientist, who will talk about modeling and animal models and cell cultures. And we'll finish it up with Dr. Amena Smith, who's a neurodevelopmental pediatrician who's just joined our staff here and was trained here by our group and works at the Moses Center for Leukodystrophy. So without further ado, I'm going to just quiet down and have Dr. Engelen take over.
All right. Well, thank you very much, Ali. Welcome, everybody. Yeah, it's my honor to give this brief introductory talk on LBSL. And basically, I'm just going to cover the disease, just the basis, so that the other presenters can then go into much more detail. The interesting thing about LBSL, like Ali said, that it is a relatively new disease. It was only described as a distinct disease entity less than 20 years ago. And the interesting thing is that even if you look back in the literature, you cannot find any earlier descriptions, which is in other diseases is often the case. And it's actually a disease that was completely described and classified on the basis of the pattern of abnormalities on MRI scans of the brain and spinal cord. So it was defined before the genetic defect was known. So to set the stage a bit for the next speakers, yeah, it's important to cover a little bit of anatomy of the brain and spinal cord, and to describe some of the tracts so that we can later better interpret the findings on the MRI scan, and that we can better interpret the signs and symptoms that people actually struggle with in daily life. So LBSL affects the long tracts in the brain, and all the way through the brainstem to the spinal cord. And these are the tracts that carry the fibers for motor function and the fibers for sensory function. And the main tracts that are affected, that are very typical, are these tracts in the brainstem and the lower tracts in the spinal cord. So how do you recognize it on MRI scans? Well, MRI scans give very detailed pictures of brain anatomy. And very simply put, they're shaded in gray. And usually on the types of sequences that we will be showing, you see that abnormal structures where there is damage will get a high signal, which is white. And you can see, for instance, here, this little white dot, that is a tract that is affected, like the tract with the red arrow also. And all these tracts that are marked have an abnormal signal and are abnormal, and the pattern is very suggestive for this specific diagnosis. And within this category of diseases, it's pretty unique that not only the white matter in the brain is affected, but also the tracts in the spinal cord. And you can see that on this cross-sectional image, you can see the white areas. Oh, excuse me, I'm going back. The white areas in the sensory tracts and in the motor tracts, and this pattern is really distinctive. And if you see this pattern, there's only very few diagnostic possibilities, of which LBSL is one. So again, here you see the pattern of high signal in the white matter of the brain, and also in the long tracts of the brainstem and the spinal cord. And what is also typical and what can help in defining the disease is that if you do a technique called MR spectroscopy, in which you can measure the concentrations of certain metabolites, certain chemicals in the brain matter, that there's a peak in lactate, and the combination of these MRI abnormalities and the lactate is highly suggestive of LBSL. But that is only part of the story, of course. If you can, although you use the MRI to diagnose it, that is, of course, not what people suffer from in daily life. And what they suffer from in daily life is mainly, especially the most typical form of LBSL, is a gait disorder. And this gait disorder is caused by degeneration of the motor tracts, the red tracts in this diagram. That you could see on the MRI scan, and also degeneration of the sensory tracts, blue on this diagram. And this combination causes people not to feel well how they position their legs and feet, and cause weakness and spasticity in the leg, and that those combinations cause a progressive gait disorder. And the abnormalities higher up in the brainstem, they cause mainly coordination problems in the arms. And what is so these typical manifestations usually have an onset in adolescence or early adulthood. But of course, as in all disorders, it's highly variable. You always have patients who do much worse or much better, but on average, it manifests in adolescence or early adulthood with the gait disorder, with stiffness in the legs and decreased sensation and the coordination problems. And cognition is usually normal. And there can be other symptoms, but they're not as prominent as what we just discussed. And what is also typical is that the disease progression is slow. It's a very slow disease with progression over years, or sometimes even longer than that, decades. So this was just a very brief overview to set the stage for the next speakers, who have much more interesting presentations. And the first speaker will go into the genetic cause of LBSL and how to further unravel the pathophysiology in model systems like mice. And then Dr. Smith will talk about her ongoing research on the clinical manifestations, the symptoms of LBSL, the progression over time, and how to study that. So now I would like, thank you for your attention, and I would like to give the screen to the next speaker.
Thank you. Thanks, Marc, and thank you, Ali, for the introduction. My name is Christina. I'm a research scientist working at the Kennedy Krieger Institute. I'll be talking about some of our modeling of LBSL, and I'm going to get into the genetics. I'd like to point out first that all of our work was funded by A Cure for Ellie. This is a family organization started by parents of a little girl with LBSL. And in a few short years, they have made connections all over the world with other families and other patients with LBSL. So thank you to them, and thank you, of course, to the ULF. And maybe my screen is stuck now. Try this again. Here we go. So DARS2 is the gene that's mutated in LBSL. And DARS2, the enzyme, is one of a family of 19 tRNA synthetases. These are mitochondrial tRNA synthetases. They assist in the assembly of proteins within the mitochondria. And I'm going to get into pretty specifically what that actually means. As many of you know, mitochondria are these dynamic organelles. They can be, they're found in every cell. There can be hundreds of them. There can be hundreds of thousands of them within every cell. So they're very important to the cell's function. They produce energy, and they're very necessary for life. So in order to understand what tRNA synthetases are, we sort of have to go back to the beginning, to the central dogma. And this is the framework for the flow of genetic information. So as many of you know, DNA is the genetic code. We like to refer to it as the recipe for all of the proteins that are in your body. And proteins are the end goal. So proteins are responsible for the structure, the function of your tissues and your organs. And unfortunately, the machinery that's necessary to make proteins can't understand the language of DNA. So we first have to turn DNA into something else called mRNA, and that's a process called transcription. And what happens during transcription is we can actually open up this double helix, and when we open that up and separate it, machinery comes in and reads that one side and sort of makes a mirror copy, and that's called the mRNA. And the mRNA is something that's understood by the protein-making machinery, and that process is called translation. So again, proteins are sort of our final goal. So if we're looking more specifically at translation, I just want to point out that we're going to work backwards this time. So proteins, this is our tertiary structure. So this is the structure of the final protein is actually really important. So if a protein isn't structured, or if it isn't complete in its 3D structure, it might not function the way that it's supposed to function. But if you unravel that protein, what you get is this polypeptide chain. So this polypeptide chain, same thing, just unraveled, and that's made up of amino acids. And amino acids are essential for life. There are 20 of them that make up polypeptide chains. They all have names, and they're all abbreviated with this one-letter abbreviation. So the sequence of that polypeptide chain is determined by your mRNA, and that's determined by that genetic code, that sequence. So the DNA to mRNA, and then machinery comes in and reads this mRNA sequence in a group of 3 that we call a codon, and then we take that sequence, and each one of those then is translated into an amino acid. So GAU becomes aspartate, which is D, and then a tRNA comes in with that D and adds it to the polypeptide chain. So that's how the proteins get made. tRNA synthetases come into play with these tRNAs, and these tRNA synthetases are enzymes. There are 19 of them, and as I said, there are 20 amino acids, so one of these actually takes care of 2 amino acids. But for the most part, they each recognize one amino acid specifically. So DARS2 recognizes the aspartate, and they also recognize one tRNA that has that unique sequence identifier. Through a multi-step reaction, the tRNA synthetase helps to put that tRNA with the amino acid, and then that amino acid can go and build that polypeptide chain. So that's how they work. I hope that made sense. So when we talk about translation and transcription, most of the time what we're talking about is this process that happens in every cell. So transcription happens in the nucleus, that mRNA comes out into the cytoplasm, and then it participates in translation. What we're talking about with DARS2 is something that happens only in the mitochondria. So mitochondria are crazy. They have their own DNA, and they have their own process of translation within the DNA. So DARS2 is produced here in the cytoplasm, and then it's actually imported into the mitochondria where it functions in translation. So all the proteins that it helps to make actually work in the mitochondria. So this is a schematic looking at DARS2, the gene. All of these blue blobs that you see are protein coding sequences. And I just want to point out that you can have mutations all over the gene. So many different mutations are associated with LBSL. And we're going to touch on this a little bit later as well. So how do we study it? We study it with different models. And two models that we use are mouse models and cell models. There are pros and cons to each of these. I'm not going to go through them. If you're interested, you can look at this later. But we've had a decent amount of success using these, and I'm going to show you about some of that data. So as I said, DARS2 is essential for the mitochondria, and it's also essential for life. So if we take a mouse and we knock out DARS2 in every single cell of the body, that's not compatible with life. So these mice don't make it through to be born. So what we have to do is we have to work around that, and we use a process called Cre-Lox recombination. And what we do here is we mate two mice together. So these mice have a sequence surrounding their DARS2 genes, and these mice have a little bit of a targeting aspect to them. So after multiple generations, we can actually knock out, we can delete DARS2 in a specific area that we specify. So we've done that, and the first time we tried it, we did it with neural stem cells, and that would delete DARS2 from every cell that goes on to function in the brain. And that happens to not be compatible with life either. So then we got more specific, and we made a model where we delete DARS2 from cells that are destined to become myelin-producing cells, and also a subset of neurons. And the animals that have DARS2 deleted in these myelin-producing cells, they're actually quite fine. So they don't have any phenotype that we can detect. The animals that have DARS2 deleted from neurons specifically, they have a very severe phenotype. So we're going to talk about that. This is a graph looking at the amount of movement that a mouse does in 10 minutes. So by the time these mice are about 8.5 months old, what we can see is the DARS2 mice, the knockout mice, they become very hyperactive. So they're moving about 150 meters per 10 minutes, which is a lot. This is a phenotype that starts fairly early. So it's something that we can detect early on, which is also nice because if we later decide to treat these mice, we can do it early, and then we can detect as they progress how this drug is working. Now, despite all that activity, these mice weigh the same as their littermate controls. But if we look at their brains, what we see is we see all this white area. And that's actually just their cerebrospinal fluid. So their brain actually shrinks. That's empty cavity right there. And you can see in this mutant picture, compared to the control, how much that size difference is. And most of that is because of cell loss in their cortex, in the cortical layer, which you can see compared there. And also, you can see a nice comparison of that difference right there, which is quantified. Now, if we look at the cells, if we look at these green cells, these are microglia cells. If you watched Dr. Watkins' talk yesterday, he called these the security guards of the brain. So they're surveying, so they're large and ramified, and they're surveying the surrounding environment. When they're activated, they take on this sort of amoeboid, this blob-like shape. And they also express markers of activation, and that basically means that the cells here are in a highly inflammatory environment. So at 9 months of age, these animals have a significant amount of neuroinflammation. If we look at them much earlier, if we look at them at 14 weeks, so way before they have any sort of phenotype, this is a graph that's looking at all of the genes. So we're looking at about 300 genes total. Every horizontal line is a gene, and every column is a mouse. So if we look at just our mutant mice here, what we see is a lot of red. And basically, that means that a lot of genes are increased. And all of those genes respond to inflammatory processes and different immune reactions. So that means that very early on, even before we have any of that neuronal loss, we're having a lot of neuroinflammation. So what we tried to do is we tried to treat these animals with a potent anti-inflammatory, and what we saw is that there was actually no effect. So this is important because we know that inflammation is not driving it. There's something else that's driving it, and we can't halt that process with inflammation. The inflammation is likely a reaction to whatever is going on with those neurons. So the important part here is that we have a model that we can use to test therapeutics. And what we're hoping to do is we're hoping to use our cell models to be able to drive the development of those therapeutics. So if we use the cells to figure out what's wrong, we can test any therapeutic that we come up with in those mice. So for the cells, we use something called induced pluripotent stem cells. These are stem cells that are derived from skin or from blood, and they're pluripotent, which means that they have no fixed developmental potential. So they can decide to be any type of cell type that we trick them into thinking that they are. These are sensitive cells. They require daily feeding. The food that they take is very expensive, so they're a little bit finicky, but we can get a lot of use out of them. So we make iPSCs by first taking skin or blood from a patient, and then we treat it with reprogramming factors. So we actually backtrack them. We revert them back to this pluripotent stage so that they no longer think that they're skin or blood cells. And then we can treat them with other factors that sort of push them towards a certain lineage. So we can then make them into heart cells, we can make them into various neurons, we can make them into pancreatic cells, pretty much anything that you want to make them into. So Dr. Ying at Kennedy Krieger has helped us to make motor neurons. So he's modified a protocol to make motor neurons in about 3 weeks. This is pretty fast for motor neurons. Not to say that it's easy, because motor neurons are very picky. Obviously, in a brain, motor neurons work with other cells, and they're in a 3D conformation, which when we plate them on a dish, they don't have. So they're not as happy as they would be in a brain, but we can sort of grow them and trick them into being happy enough that we can test a lot of their functions. So this is what they end up looking like. So the green is just looking at their structure. The blue is the nucleus. That's kind of like the headquarters, the brain of the cell. And if we look at the DARS2 protein, what we see is that in our LBSL patient, I'm showing just one patient line here. We have a couple others in the lab that we're working on confirming results for, but the data that I'm going to show you are from one patient. We can see that the DARS2 protein is decreased both in the iPSC stage and when it's a motor neuron. So overall, it looks like the motor neurons require less DARS2, but that pattern for the LBSL is consistent. Now, this is still enough protein, we think, to function normally. And because we have a reasonable amount of DARS2 protein, what we wanted to do is we wanted to look at all of the other proteins in the mitochondria. So we learned that DARS2 participates in the production of proteins within the mitochondria. So are those really affected? And this is kind of a complex figure, so I'm going to walk you through it a little bit. But every dot that you see is a protein. And then proteins interact with other proteins. Proteins might function together in a certain process or be located in a similar part of the mitochondria. And if they interact, if they work together, they're going to be connected with a blue line. And then what I want you to pay attention to is this halo color. So a blue halo around a protein means that that protein is decreased in LBSL compared to control. If it's red, that means it's increased in LBSL compared to control. And then if it's gray, that means that it's unchanged. So what we see here is pretty complicated, but these proteins all kind of work together. This is another family of proteins. And then we have some kind of loosely connected groups here of proteins that all work together. But what I want you to notice is that the blue and the red halos are not confined to any one region, any one function, any one part of the mitochondria. So they're all kind of spread out and diverse. And there aren't really that many of them as we would expect if it was having an effect in the translational process. So what we take from this is that the cells are stressed, but it's not necessarily related to translation. So there's no evidence that mitochondrially translated proteins are affected. And we can also look at the amino acids. So DARS2 adds that aspartate. And if it wasn't functioning properly, we would assume that other amino acids are being put in there in its place. And that doesn't seem to be the case. So what's next? So next we want to look at just how these cells grow, how they develop, and how they function. So to do that, we use a machine. This one's called an IncuSite. And this is a really nice camera that sits inside the incubator. And we don't disturb the cells at all, but we can take really nice pictures of them about every hour for a couple of days. And when we do that, we end up getting a pretty nice video. And from this video, we can look at the number and the size of the cells. And then we can look at the processes. You can see that these cells grow processes, which they should. And then we can measure those processes, both the number and the length. So some of these neurons, you can see, grow really long processes here. And that's normal because neurons need to communicate. But when we quantify that, we see that the branching, so the number of those processes and the length of those processes, is less in both cases. So these cells seem to appear normal and they grow fine, but they seem to have fewer branches and those branches are shorter. So this is really important for understanding how these cells function and for narrowing down what might be wrong. This is our technician, Brett. he has kind of made this his quarantine project. So what he's working on is developing a program to be able to track these cells. So he wants to look at the distance traveled of these neurons, and we can do that by tracing them over time with those same videos, and then quantifying both the control and the LBSL to see if the LBSL cells travel as far, if they make these connections, and if they clump behaviorally or as a normal neuron should. So that's hopefully something that we will have some more data on soon. And how do these neurons communicate? They communicate through action potentials. So Dr. Min talked about this a little bit yesterday, but basically these cells are electrically excitable. So they communicate between cells with neurotransmitters and chemical signals, but then they produce this electrical signal that we call an action potential. So this is a all or none, it's a voltage change within the cell, and the action potential is generated, and then that signal gets passed down the axon to the end of the cell where it can be sent to another cell to get an action potential. And you can see the myelin here, which facilitates the signal, the processing of that signal down the axon. And if we plate these neurons on right on top of electrodes, we can actually measure the population change. So if a group of neurons are all responding, they're all going to form these action potentials, and we'll be able to pick that up on these electrodes. And you can see the plate here, kind of the size of what this plate looks like. And if we do that, what we see is that there are actually fewer action potentials that they form. So over multiple days, we measure these cells and they fire, but the frequency of that firing is less. So over 30 days, you can see that they have the same general pattern, but they fire fewer times. So what's great about this assay and the IncuSite, the video assay, is now we have two means to measure different therapies. So if we were to give a drug to these cells somewhat early on, we could see if we could change the trajectory of that cell behavior to act more like the control cells. So we have a couple different systems to measure those changes. And one therapy that I just want to touch on is antisense oligonucleotides. So this is something that Dr. Vanderknap's group has proposed a couple years ago. They had a nice experiment in one of their LBSL papers with it. Dr. Fatimi talked about it a little bit yesterday. But why this is good is because many of our patients, or most of the LBSL patients, you can see hopefully in these numbers here, most LBSL patients have a mutation right in this area, intron 2, which is right in between these two protein coding areas. And what happens with that mutation is it leads to the skipping of one of those exons, and that will alter that protein structure. And what Schertzy in our lab is working on doing is designing these sequences that can go in and bind to that area in between and sort of cover up that mutation. So if we can cover up that mutation, when the protein machinery comes in to read that, what happens is it'll hide that error, and then we'll get the full-length protein. So that's something that we're currently testing, and we're going to use both of these assays, both the action potentials and the video IncuSite system, to test the efficacy of those therapies. So that was quick. I hope that wasn't too fast, but the conclusions: DARS2 is essential for life. It's essential for neurons. And then the neurons, we have a couple different ways to show functional deficits in these cells. So altogether, we have a couple nice model systems to test therapies and to continue to get at the effect of these DARS2 mutations. So with that, thanks to the lab, obviously, to the ULF and A Cure for Ellie, and then I'll pass it off to Dr. Smith.
Okay, great. Thank you, Dr. Mertz. So hello, I'm glad that you could join us today. I'm Amena Smith-Fine. As Dr. Fatimi mentioned, I'm a neurodevelopmental pediatrician working in the Moser Center for Leukodystrophies at Kennedy Krieger Institute. So I work very closely with Dr. Fatimi and Dr. Mertz, who just presented. And we've also recently started a collaboration with Dr. Marc Engelen and Dr. Maria Vandernap at the VUMC Amsterdam. Dr. Engelen had presented first. So today I'll be giving you an overview of what we've learned from our ongoing natural history study in LBSL. And then really an exciting aspect of this project is that we're using wearable technology to get information about walking and balance in LBSL during teleresearch visits at patients' homes. Just a second. Okay, so why start a natural history study? First of all, a natural history study is a collection of health data repeatedly over time to understand disease symptoms and outcomes of a disorder and how that progresses. There are a couple main reasons why this type of research is important. First, we at this point know enough to realize that LBSL progression might look different depending on age of symptom onset. So generally, earlier age of onset, for instance, in the infant age, is associated with earlier loss of independent walking and earlier wheelchair dependence. But beyond that, we really don't know what factors predict how LBSL symptoms will look over time. Possibilities are things like the combination of DARS2 mutations a person has, or the burden of white matter changes on their brain, or spinal cord MRI. But this has been tough to determine especially because the disease is so rare. The other main reason this is important is that we don't yet have a cure or really any FDA-approved treatment specific for LBSL. Although, as Dr. Mertz mentioned, there are a number of different possibilities in the early drug development pipeline. So, still working on a cure, and the natural history study basically allows us to get a baseline understanding of disease symptoms and outcomes so that we can compare, we'll have something to compare the results to when treatments are developed and we get to that clinical trial phase. So simply stated, then our main goal is to establish clinically meaningful quantitative outcome measures for clinical trials in LBSL, which would be our next step. So our approach has been to get information about gait and balance using wearable sensors and a clinical ataxia scale, which I'll talk about shortly. And then also we're getting information about clinical history and neurologic exam information, information about cognitive abilities that might impact school or work performance in LBSL, and also DARS2 mutation reports. And this is important to know if certain mutation types or locations on the DARS2 gene influence the severity of disease symptoms or the rate of symptom progression over time. So we're doing these basically remote web-based assessments during virtual visits at patients' homes. And this is with the intention to increase participation in the study and decrease burden of travel on people. And then importantly, we're actually recruiting both LBSL patients and age and sex matched healthy volunteers, since the walking and balance variables do differ slightly based on both of those factors. And we are currently repeating assessments every 6 months over a full period of 5 years. So we chose the OPAL system of wearable accelerometers for our study. This is a non-invasive, relatively inexpensive technology for recording movement in a real-world context. They've already been used to measure disease-specific features in multiple sclerosis, Parkinson's disease, cerebral palsy, and they collect similar information about gait and sway that have been reported in other ataxic disorders. So the system consists of these 3 watch-size battery-based accelerometers, sort of looks like a Fitbit, and they submit a synced signal to a central sensor through very low power radio frequency waves as the tests are happening in real time. So we put the sensors on the feet and waist of the participants for a series of brief walking and balance tests, which I'll talk about shortly. And then for the virtual visits at home, we send an OPALS kit pictured below, a web camera that can be controlled remotely, a laptop, and then a device manual which has, in simple language, all the instructions for how to set up the tests. And so we do this through Zoom, which most of you might be familiar about at this point, and can do remote takeover and essentially guide all of the testing. So it's pretty easy, and most people who participate are actually able to set up the equipment before the scheduled call. So we selected the scale for the assessment and rating of ataxia, or the SARA scale, to measure ataxia in our participants. So this is a brief and sensitive tool that consists of 8 different exam maneuvers shown below that are commonly tested by a neurologist to check for signs of cerebellar or sensory ataxia. The SARA scale has been used to look at disease severity in spinocerebellar ataxia and Friedreich's ataxia. And in these disorders, it's shown very good reliability between 2 tests done in a short period of time and between different examiners of the test. So unfortunately, the SARA is somewhat limited in that it was able to show progression in spinocerebellar ataxia after a year, but it didn't capture all ataxia symptoms reliably at disease onset. However, a wearable sensor measurement of gait ataxia was shown to be superior to the SARA in the early stages of spinocerebellar ataxia. So we're essentially exploring the use of both of these measures to look at disease severity and progression in LBSL. So within our larger group of patients enrolled in the natural history study, today I'm going to focus on a group of 8 patients who are able to walk and follow test instructions consistently. So among this group, the median age is 10 years, so pretty young, ranging from ages 5 to 23. There are about 40% females and 60% males involved in the study. The mean age at disease onset for them was 4.5 years, but there was a range from 9 months to 16 years of age. And the presenting signs pretty much universally were ataxia and fine motor delay. An interesting thing that we found so far is that 75% of patients reported a brief worsening of their motor skills and sometimes some cognitive sluggishness, basically when a fever, illness, or minor injury or minor head injury happens, and that typically resolves after a few days to a week, but is recurring. And then gross and fine motor ability scores for our patients were really in the mild to moderately impaired range of 1 to 2. Both the gross motor and the fine motor ability scores are on a range from 1 to 5, with 5 being the most severe impairment. And then their SARA ataxia scores were also in just the mild to moderately impaired range from 1 to 16. And this is on an overall scale of 0 to 40, with 40 being the most severe form of ataxia. So breaking down our ataxia scale subscores a little bit more, essentially most patients, nearly 90%, had involvement of the lower extremities. And so this is reflected in the stance, the heel shin slide, and the gait scores. This was followed by ataxia symptoms affecting the arms and the trunk, with a hand movement sitting, finger, nose, finger, and finger chase tests. And then only about 40% had mild speech impairment. And so essentially this finding of the lower extremities being predominantly involved is consistent with other published reports in LBSL. So we asked about school performance and supports needed, which of course was retrospective for our adult participants, and found that while the prevalence of a diagnosed learning disability was low, 50% actually reported mild cognitive complaints. Is typically just related to attention or processing speed. And 63% actually had a current or past history of writing problems. Overall, 63% of patients required either formal 504 or individualized education plan supports in school for these problems. A chart review of neurologic exams showed us that nearly 90% of patients had sensory deficits in the feet or the ankles, reflected as impaired vibration sense and a positive Romberg, followed by ataxic gait, low tone, with or without spasticity at the ankles, increased reflexes, mild weakness, and then about 50% had impaired coordination of arm movements. The eye movement abnormalities were rare, occurring in only 2 people, and 1 patient had hyperactivity on mental status exam. So transitioning to our findings with the OPALS testing, for the 2-minute walk test, the participants walk back and forth between 2 points on a 22-foot walkway set up on a clear path in their home, usually the kitchen or the living room, for 2 trials. So a gait cycle is the period of time from the foot's first contact with the ground, this is usually a heel strike, until that same foot makes next contact with the ground again. So the OPAL sensors collect about 16 variables during each gait cycle, which encompass all the different phases of where the foot might be positioned in space. I'll just be showing a few of those variables today. And a lot of information is collected. Basically, one walk trial gives us about 20 to 40 gait cycles. So I'll just show a video. This is a 10-year-old girl with a moderate SARA score, just as an example. So you can see a little bit of stumbling as she walks subtle, and some mild hesitancy with turns as well. Okay. So the stride length is the forward distance traveled by the foot during a gait cycle, and a short stride length is a compensation for unsteady gait. So as we hypothesized for our patients with ataxia, the LBSL group had a shorter stride length compared to their age-matched controls. Although this difference wasn't statistically significant in our small group of participants, meaning that there was a greater than 5% chance that the differences were due to random chance alone. The time that it took to complete a gait cycle in seconds was just slightly longer for the LBSL group. And then finally, the LBSL group had a slower stride velocity in meters per second compared to the control group. And this is important because per the literature, this difference of about 0.1 meters per second is actually clinically meaningful. So the hallmark sign of ataxia, of course, is variability in stepping and in strides, which the OPALS estimate by the lateral step variability. So it's a little bit of a complicated concept, but basically when you consider 3 consecutive foot placements made by the same foot, the right or the left foot, this is the variability of the perpendicular deviations of the middle foot placement from an imagined line connecting the first and the third. So basically, this indicates a larger lateral step variability indicates a veering or a stumbling gait, and larger deviations might also indicate a wider stance base to preserve gait stability. So we found for our LBSL group, there was a greater lateral step variability versus controls by about 2 centimeters on average. So circumduction is a lateral swing max, or the maximum amount that the foot travels perpendicular to forward movement during stride, and positive values indicate more movement to the outside. So we found our LBSL participants showed greater leg circumduction than controls, but this was with a large degree of variability. Circumduction is most often associated with leg weakness or increased tone in the legs or the ankles. We next looked at the height of the foot at mid-swing relative to its start position while standing, and found that our LBSL group average was higher than the control average. So this is also called a high steppage gait and has been reported in disorders with cerebellar ataxia, peripheral neuropathy, or spinal cord lesions. And it's thought that the high step is probably a compensation to allow the foot to clear the floor during a stride. So when thinking about turning during the walk trials, we found that our LBSL participants took slightly longer to turn. They had a smaller turn angle, and overall this resulted in a slower turn velocity in degrees per second compared with controls. So together, these findings are indicating that there's some balance and stability as they negotiate turns. So our remote balance testing consists of 4 different postural sway conditions in which we challenge participants by either removing visual input, this is asking them to close their eyes, or narrowing the base of support by putting their feet together. So they perform 2 different trials of each condition. The stance width is standardized with a foot plate, and the sway is measured from the lumbar or waist sensor pictured here. The OPALS collect about 10 key variables in different planes while these tests are occurring. So I just wanted to show you some more videos of our same patient. So this is her, her sway with feet apart and eyes open. Feet apart and eyes closed, which is a little bit more challenging. Feet together and eyes open. And then feet together and eyes closed, which is clearly the most difficult position for her. So shown above are plots mapping this girl's sway path here on the right compared to an age-matched volunteer on the left. This is a view looking from the top down. And so you can easily see that across all conditions, there's greater sway for the LBSL participant, but especially in the 2 conditions where her eyes are closed, and it's greatest when her eyes are closed with a narrow base of support. So here are the summary sway plots for the control and LBSL groups, reflecting really a similar trend. And below, we've quantified the sway area for the groups. So as in our representative patient, the sway area is certainly greatest in the feet apart, eyes closed condition and the feet together, eyes closed condition. And then to further evaluate the impact of removing visual input on the sway, a Romberg quotient was calculated for each person. So this is defined as the sway area with the eyes closed divided by the sway area with the eyes open. And we found that our average Romberg quotient was significantly increased for LBSL subjects, which likely reflects proprioceptive contributions of the dorsal column pathway fibers in the spinal cord. So essentially, our takeaway from this is that the spinal cord white matter changes seen in LBSL may be really an important contributor to the overall motor disability experienced by persons with LBSL. So lastly, these plots are just demonstrating that our SARA ataxia score is strongly correlated with the lateral step variability during walking. You can see a nice line here, and this was statistically significant. And then also the SARA ataxia score was correlated with the sway area during standing. And this basically supports the idea that the wearables are capturing variables that are clinically meaningful and represent ataxia. So in summary, through our virtual testing at home, we found that our group of LBSL participants with mild to moderate disability walk and turn slower, have shorter stride lengths, and show more gait variability compared with healthy volunteers. The lateral step variability and sway area measured by the wearables is strongly correlated with a clinical ataxia scale that's widely used. And this is likely related to cerebellar or proprioceptive ataxia, or sensory or cerebellar ataxia. And then finally, removing visual input has a greater impact on the sway area than narrowing the stand space, suggesting that possibly the dorsal column dysfunction in the spinal cord is an important contributor to instability while standing. So our next steps are to perform advanced white matter tract MR imaging sequences to further understand how changes to the white matter tracts in LBSL are contributing to impaired gait and balance as measured by the wearable sensors. We are in the process of determining how useful the OPALS remote testing is over time to get in quantitative gait and balance information that can be used as outcome measures for clinical trials. And this is by repeating testing every 6 months. So this data showing what things have looked like over the first couple of years will be presented at the November 2020 LBSL conference hosted by the Kennedy Krieger Institute. We are currently, actually since last year during Big ULF, we've been partnering with Drs. Marc Engelen and Mario Vandernap at the UMC Amsterdam to recruit European LBSL patients, which we're very excited about. And their efforts hopefully will be starting very soon, later in the summer. And then finally, this work really could not be done without the commitment of our LBSL patients and families. We really appreciate everyone who's been involved in the study so far, but it is also really important for us to recruit new participants. So if you're interested, I have the contacts below for myself and our research coordinator, Jordan Goodman, for more information about how to join our study or really for any questions that you might have. And my understanding is that the presentations are recorded, so you'll have this to reference later. So just want to thank you for your attention and participation today. Again, especially a shout out to our LBSL participants and families. Thank you to Dr. Ali Fatimi, who is the principal investigator of our project and his lab at the Moser Center for Leukodystrophies at Kennedy. Dr. Amy Bastian and her group at Kennedy has been really instrumental in advising us on the use and interpretation of the OPALS data. And also a big thank you to the McGinn family and A Cure for Ellie for their support in many ways, not in the least including financial support and helping to really spread the word and get people excited about our study. Our other funding is listed below. So since there's like a little bit more time, I also, and you'll be able to reference this later, I had also included a couple of papers by Dr. Vandernap's group and our own group that might be helpful for more overall clinical information about LBSL, and the Cure for LE website and the NORD website, which is also just a good resource. Thank you very much.
Okay, maybe we can actually all of the speakers on. That would be great. Thank you for these talks. I thought it was great. Might be biased. I thought maybe we'll try to answer some of the comments that have been made. So, for one, I think a general statement is that, like we just discussed, this is a recently discovered super rare disorder, and there is many individuals that have relatively unique symptoms, and therefore, you know, while the symptoms that you heard about, like the neurological symptoms, are relatively unifying, other things tend to be quite unique, and so whether they're truly related to the disorder or not, we really don't know. So, for instance, I'm going to start off with the question that was raised about diet. In general, we have not noticed so far that you know, these individuals have a greater appetite or less appetite, or that have any weight issues. There are certainly individuals on being smaller, but I'm not aware of actually people having excessive weight. So I see that one comment coming, I think, from Ms. Schneider. You know, in terms of mitochondrial disease in general, it's actually more the other way around, that most mitochondrial patients, if having weight issues, are on the smaller side, they have failure to growth. That's a very common problem in mitochondrial disease in general, but not so much in LBSL. Similarly, in terms of other symptoms, I saw a comment about somebody gotten worse after pregnancy. You hear that often in neurological disease, where people get worse after pregnancy. Part of the problem is that whether you can actually find it and say this was truly related to pregnancy, or whether it's just that, you know, as an adult person, the symptoms have gotten worse. What we do, and we don't have any other patients, as far as I know, who have this disease who went through pregnancy. Certainly what we do know is that it seems that this disease is a rather episodic disorder, meaning that patients tend to be pretty steady, and then oftentimes it's an infection or some sort of an infectious illness or an inflammatory illness that results in acute decompensation of the neurological status. Now, we've seen many of these people recovering, and we don't know exactly if that recovery is sort of completely spontaneous, or whether it has been assisted by the things that you've been trying to do in general to help the patient stabilize. But we've certainly seen patients going through an, you know, viral illness or influenza or similar illnesses, and then after that having a sort of decompensation. So it's actually many times the first way these kids present is that they're fine until they are like in the late first year of life, where kids, you know, often go to school and start getting exposed to viruses and get sick, or may even have had some form of a vaccine, and it seems that they're getting sick. And so as a consequence of this, there are certain things that we tend to do for general management. We do think, for one, for instance, that vaccinations are really important. We do not say do not get vaccinated. We say get vaccinated, because vaccines are there to protect you from really serious illnesses. And while you might say, oh, the vaccines triggered some inflammatory process, the consequence of having the actual disease levels. So that's an absolute sort of must in patients. Trying to prevent serious infections as much as we can is an absolute must. Along with that, it's also important to say that there is certain things that you want to do, like in other mitochondrial diseases, to protect yourself. So we want, for instance, children to have as little as fever as possible. So if they get sick with an infection, We try to make sure that the fever is controlled tightly with, NSAIDs, Tylenol, and, acetaminophen basically, or Advil. We are quite proactive on keeping kids hydrated. So if somebody has diarrhea and vomiting many times in a normal child, you will call a pediatrician. He's like, well, I'll try to keep them at home, try to just spoon feed them slowly. Here we are more proactive. Kid gets dehydrated. We don't wait for too long to bring him in to give them actually an IV infusion so that they remain hydration. And they remain their electrolyte balance. At the same time, we are relatively proactive in treating them with antibiotics. So while many times we recommend against use of antibiotics with common viral infections, here we don't want to take the risk and are more proactive on prescribing antibiotics for throat infections and so on. At the same time, there is also some concern that maybe head injury may have a concern on an effect on the disease. There is no clear data to prove that. I'm generally not a great fan of contact sport personally for my own kids. I would say that we advise against contact sport, but we certainly want the kids to be engaged. We want them to be active. This is a very slowly progressive disorder. These kids should have the best possible life as possible. They should enjoy activities outdoor. They should engage in sports, with the understanding that they're going to have gait issues. And given that they have gait issues, we want to make sure that they're careful. So if they do something but they're at high risk of falling, that's probably not a good idea, or they should wear a helmet. So these are sort of the general precautions that we take with this disease. Let's see here. Is there other questions and other people who want to maybe chime in here from the speakers?
So I guess we skipped. Do you want to go ahead, Marc? Oh, I guess we skipped how the gene is passed. Someone had a question about having a different mutation on each copy of the gene. So patients are what we call compound heterozygotes. So they get, so both of their parents would be carriers. So each of their parents would have one copy of the DARS2 gene that has the mutation. And so they pass their mutation to the child. Both parents do. And in that case, because there are so many different mutations, then the child ends up with two different mutations. So just because there are so few patients that we're aware of, and there are so many mutations, it's really hard to draw any sort of conclusion about what's pathogenic and what's not, or if there's any sort of severity associated with a particular mutation. So it's really early on to answer those kinds of questions.
I noticed a couple of people saying, oh, one was a known mutation, one they weren't sure. That's almost uniformly the case with most rare genetic diseases, is that we oftentimes know of a mutation already, and then the other one is something that we hadn't seen before, which doesn't mean that it's not disease-causing. That just means that we just hadn't seen it before. There is also, in terms of passing it on, I see a question. One person says, my wife is diagnosed with LBSL. Should we screen my child? She does not have any symptoms. No, so when a person who has LBSL has the actual disease, has both genes mutated, and by default will pass on one of those two genes to the next generation. So the child will be a carrier but will not have symptoms. If that child then has another has an offspring with another carrier, then they have a risk of 25% for each pregnancy that child will be affected. So your grandchild could have LBSL if your future child or your current child, who's just a carrier, just happens to, mate with another carrier, which is very, very, very unlikely, right? So, I mean, I know it happened already with these families here, but the disease is super rare. So we're currently not recommending sort of screening for who are carriers in general. But yes, the child will be a carrier, but will not have. There was a thought a comment about one individual, Miss Black, stated about their child who had sounds like a severe form of LBSL. We didn't really talk about that, but there are individuals that we know of that are having a more severe presentation of this disease that's almost they are born and they often have a small head already right from the get-go at birth, often have low tone right from the get-go, and tend to have epilepsy and tend to have intellectual disability. So those are more severe cases that are rarer and don't follow really the pattern of disease like in the classical form. So yes, we are aware of those cases. And we think that they may have something to do with the mutations, although we can't prove that for sure right now. What we do know is that when we knock out the gene, as Dr. Mertz talked about it, when we knock out the gene in the neurons in mice, their behavioral, they have very severe behavioral abnormalities. Their behavior is very abnormal. So certainly, if you miss the gene, it's not compatible with life. We think that there is still some leftover function of the gene, and it's probably less so in the more severe cases. But we can't prove that in people yet. That's one of the things that would be very interesting in studying the cells of individuals. So, for instance, if you could get the cells or blood samples from your children, that would be very helpful here.
All right, thank you so much for our presenters. Thank you for your time and answering everyone's questions. And thank you to all of our people who are watching on the YouTube side. Hopefully we will be all able to see each other all next year.
Great. Thank you.
All right.